I added that. Yeah, I read that it's still good.
Okay, hello everyone. So we're gonna get started. Nice to see everybody lively today. Feeling alive. Okay, so we're gonna pick back up with our understanding of how the kidneys reabsorb water. So this is part of the beginning of that topic, partway through where we left off on Friday. So hopefully we understand, right? We have begun to think about the fact that water reabsorption is one of the main roles of our kidney. So we're filtering blood, we're reabsorbing water, we're maintaining blood volume as we do this. So this is one of the major ways in which long-term we can regulate blood pressure by regulating how much fluid is in the blood plasma. Fluid creates pressure on the walls of its container. So we already know that a lot of water is getting reabsorbed in the proximal tubule. So we trace through that. That's not regulated. We're beginning to understand how we're reabsorbing water later on in our nephron. Okay. So we thought again about osmosis. So we know that water diffuses down its own concentration gradient, but usually we're looking at solutes. So we can think about water as being pulled towards solutes. So pulled towards things like ions, right? As water moves to go water down the area where we have a high solute concentration. So in this way, water is often going to follow solutes that get moved through a space across a cell wall or cell membrane. So that's what we're going to see today as we start to talk about the medullary osmotic gradient and how it gets set up. So the medulla is that central part of the kidney. So if we're drawing a kidney, right, we're thinking something vaguely that shaped. So it has a cortex, meaning an outer shell, and that has a medulla, which is the central part, arranged in renal pyramids. Okay, so those pyramids are really where we have the medulla. So you can actually see those if you cut a kidney open. Okay, so they're going to have different properties, and specifically, these pyramids, the medulla, are going to have a different osmotic gradient, different osmolarity, so a different pull on water than we see in the cortex. So we're going to trace through today how that difference occurs, like how that gets set up. So we are going to walk through the whole process in order to understand the different properties of the parts of the nephron that we're tracing through today, okay, which is mainly going to be this loop of Henle or the nephron loop. Okay. If you get super lost, the most important part to know of this process is the endpoint. So we're going to be tracing through how we get there. But what I really want you to know is what our steady state in a normal human is in a normal, fully functioning kidney. Okay. So when you come back to this, if you need to triage and prioritize, focus on that. Okay. All right. So we're thinking about establishing what we call the medullary osmotic gradient. So medullary, because we're thinking about the medulla. Osmotic, because we're thinking about how the medulla pulls on water. So this is going to be key to water reabsorption, the pull of these solutes on the water in the filtrate flowing through the nephron. We're thinking about a gradient because we're going to see that as we dive down into the medulla, we don't have a steady single number that we can remember as the osmolarity of the medulla because it's getting stronger. Our numbers are getting higher as we go deeper into the medulla. So that's the gradient part, the difference, right? So we're seeing essentially an increase in the solute concentration as we go deeper down, okay? So that's why it's a gradient. A gradient is like a hill, basically. Okay. So what's happening here is in our cortex, this is steady state, right? So we have a osmolarity of 300 here in the cortex. So that's in that outer shell of the kidney. And that's where we've been thus far. That's where you'd find our afferent and efferent arterioles, where you find the glomerulus, and where you find that proximal tubule or the proximal convoluted tubule. Those are equivalent terms. Okay. So that's where we've been so far. Now we're diving into the medulla, though. We're going to start out at 300, but we're going to end up with an osmolarity that is much, much, much greater as we dive down deep. Okay. This means that we're going to exert a progressively stronger pull on water in the filtrate, as we dive down deeper into the kidney. Okay. So that's our medullary osmotic gradient. It's going to be important both as we have filtrate or fluid traveling through our tubules, but then also we have our collecting duct diving back down into the medulla. So while it's not involved particularly in setting up this gradient, so what we're about to talk about, it is going to be affected by this pull. Okay, so any fluid remaining in the collecting duct is getting pulled by this medullary osmotic gradient as well. Okay. So overall, the osmolarity of the interstitial fluid, so the fluid between the cells in the renal medulla, so in these pyramids, is varying based on how deep in the kidney you are. We have a lower osmolarity near the cortex, so up, and a greater osmolarity near the renal pelvis, so deeper down. Okay. All right. So this is the result of something called the countercurrent multiplier. So countercurrent, counter meaning opposite, current meaning a flow. So the countercurrent multiplier referring to the fact that in the loop of Henle or the nephron loop, so this sort of turn that I've been drawing out kind of sketchily, okay, we have fluid flowing in a specific order, right? It flows down into the medulla, and then it flows back up and out, back towards the cortex before eventually diving down again. So this is a countercurrent multiplier because we have opposite directions, down and up. So that's all that this term is referring to, really. So that we have fluid flowing in opposite directions. Different things are happening in the descending and ascending limb. We're basically multiplying the effect of this pull on water. So when we start out, we're going to start out in our cortex. So our pull on water, our osmolarity of the cortex is 300 milliosmoles. Okay, when we're starting out. Okay. This is also going to be how we start out with our osmolarity of the fluid flowing through the cortex. But by the time we exit our loop of Henle, we're going to end up with fluid in the distal tubule that is lower in osmolarity than the cortex. Okay. All right. So when we're in the distal tubule, we're going to be down to fluid that's at 100 milliosmoles, still surrounded by tissue that's at 300. Okay. I'll draw on this as well. Okay, so we're starting at 300. We travel through this loop, dive deep down into the kidney. We're going to see lots of changes in osmolarity as that happens. More changes as we go back up, ending in the distal tubule with an osmolarity of 100 in a renal cortex with an osmolarity of 300. And this is why eventually we're going to see that as we regulate water reabsorption, this difference is going to mean that we are able to pull on the water in that fluid in the distal tubule, okay, because water is going to flow towards the higher number. Okay. Right. So now we're going to take a look at some of the features of the loop of Henle as we start to understand what's going on in this loop in the renal medulla. So this is showing us our cortex. Okay, so our cortex, this outer shell, would be at 300 milliosmoles. The osmolarity of the kidney on here is not labeled. We're just tracing that osmolarity, all these numbers of the fluid. Okay, so we're going to see that we're starting with those 300 milliosmoles. We just saw that we're going to end with 100 milliosmoles for that fluid. As we go down through the loop, we're going to see we have increasing osmolarity, and we'll take a look at why that happens. And then as we go back up, okay, we're going to see decreasing osmolarity in that fluid. Okay. Decreasing until we get all the way to 100. So this is going to be due to different properties, essentially of the cells in the descending and ascending limbs of our loop of Henle. Okay. So in our ascending limb, so the second half, the second half of this loop, okay, so where we have our arrow going up, where we have this osmolarity decreasing as the fluid travels forward, our tissue is not permeable to water. So water is not able to cross the membrane of this ascending limb. Okay. The role of the ascending limb instead is to increase the solute concentration of the renal medulla that surrounds it at the same time as it's decreasing the osmolarity of the tubular fluid or the filtrate traveling through. Okay. So the way it does this is with transporters. Okay, so this number 1 here, what it's showing us here is that we're taking solutes, so chiefly sodium and chloride, also some potassium. We're actively transporting them out of the fluid and into the medulla. Okay, so when we take a solute out, right, so we're dropping the concentration of solutes in the fluid, and this is why this number is dropping, right, because we're taking this out, right, and at the same time, that solute is ending up somewhere. Where it's ending up is the renal medulla. So this is essentially increasing the osmolarity here in the renal medulla. And we'll see this labeled on in some diagrams in a second. Okay. So that's all happening in the ascending limb. So this is active transport, just meaning it takes energy to run these transporters. Okay. In the descending limb, so the part where the fluid is traveling first, so we have our descending limb starting with fluid at 300 milliosmoles. We can see that it's increasing in osmolarity here, right? So this number of the fluid is getting higher and higher and higher. That descending limb is permeable to water. So permeable, meaning water can cross the membrane, right? Permeable to water, water can cross that membrane, okay? So what's happening here is that we're going to see that the osmolarity of the surrounding medulla, as it's getting higher and higher and higher, as we go down towards the renal pelvis, is exerting a pull on the water that we have here in the fluid. Water can cross these walls. It can't cross the walls in the ascending limb, but it can here in the descending limb. So the water travels out of the fluid here. As we have water travel out, that's essentially concentrating the solutes that remain in the fluid. So the osmolarity of that tubular fluid is going up and up and up. Water is coming out. And because we have capillaries surrounding this, this doesn't have too much of an effect on watering down the medulla because that water then goes into the capillaries. So that is the big picture view of what's happening here. So ascending limb, impermeable to water. Its role is to actively transport solutes to increase the osmolarity of the renal medulla. The descending limb, which happens first, is where we have tissue that's permeable to water. Water can travel across it, so we have some water reabsorption happening in the descending limb. Okay. So we're going to look at this whole diagram in detail. This is all just the setup for this final step, which is our steady state system. So when I said, if you get lost, just remember our endpoint, that's what we're going for. Okay. So what we're looking at is kind of a theoretical setup, how we create a kidney that has this osmotic gradient. So start out with number one. So when we begin with our nice fresh kidney hasn't been doing anything yet, but we've formed a kidney. Okay. We have a kidney that has the same osmolarity everywhere. So our beginning kidney, just everywhere, everything has an osmolarity of 300 milliosmoles. Okay, that's our base value. Okay. So before we start running the active transporters in the ascending limb, we'd be at equilibrium everywhere. There'd be no particular pull on water because we don't have a difference anywhere. We don't have a gradient anywhere at all between our tubular fluid and the tissue of the kidney, right? Everything's just the same. Okay. So that is not the state in your normally functioning kidney, but we got to get there. So the first thing we're going to do in order to start to change this situation is to actively transport some solutes out of the ascending limb. So the ascending limb has transporters that use energy in the form of ATP to move these solutes out. So sodium, our friend, chloride of potassium as well, moving into the renal medulla. Okay. So as they move into the renal medulla, you can predict what's going to happen to these numbers. Give me a second. Okay. So we're going to be increasing the osmolarity of interstitial fluid. Okay, so we'll click forward. And here's our new situation. So once we've moved these solutes into the renal medulla, we're not at 300 anymore. So everywhere we moved solutes, we'll go up to, say, 400, right? However, this also had an effect on the tubular fluid, right? So we increased the osmolarity of the renal medulla because we moved solutes. But if we have an increase somewhere, we're going to have a decrease on the opposite end, right? So this active transport also had the effect of decreasing the osmolarity in this tubular fluid. Okay. Right. So now we start to see some differences. Right. We can look at many places actually now that we see differences. So we can see that we've changed the osmolarity of the descending limb versus the ascending limb. We can see that we've also changed the osmolarity of the fluid in the descending limb versus the medulla that it's traveling through. So an important thing to note here is that this fluid, this tubular fluid at 300, right, is less than this 400 value that we now have in the renal medulla. So this means that we're exerting a pull on the water in the fluid. So that 400 is pulling from the 300, it's pulling water. We think of high numbers of osmolarity as exerting a pull on water towards them. Okay. So we're now going to see water moving out of the descending limb. Okay. So water is being pulled by that greater osmolarity. So it wants to move out of the descending limb. It can, because of the features of the cells in that lining of the descending limb. So even though there's a difference between the ascending limb and the medulla, water can't flow out of that ascending limb because it doesn't have the right features. Those cells don't have the ability to allow water to cross. But in the descending limb, we do. So the descending limb starts to have movement of water. So now we see the descending limb come to equilibrium with the surrounding tissue. So we have water move until both of these are at 400, right? They're matching. So we would refer to them as isoosmotic because they have the same osmolarity. So same osmolarity, no osmotic difference between our descending limb and the medulla, but we do still see that there is a difference between the fluid in the descending limb and the ascending limb. Okay. So next, we want to think about what's going on with the fluid. Okay. So we're not just having fluid just sitting still in the tubule. Fluid is actually moving forward, right? We constantly have blood flow going through the afferent arteriole into the glomerulus. All the constituents of that plasma, all the things that make up that plasma, except for like proteins and blood cells, moving into Bowman's capsule, right? So we looked at our net filtration, we looked at our glomerular filtration pressure, glomerular filtration rate. So we have fluid moving and pushing forward. So that's what's going on in the diagram here. So what we're looking at now is that fluid moving forward. The fluid in the cortex is just at that same value, that 300 milliosmoles, as the surrounding cortex. Right? So we're pushing forward fluid with this lower osmolarity, right? It's physically moving down into that descending limb. So we push down this fluid that was previously in the cortex, and I'll circle them in the same color. So the fluid that was previously in the descending limb is now pushing forward as well. And so we just pushed it on up, okay, partially into the ascending limb. And then the remaining stuff is fluid that was in the ascending limb before. Okay. So we pushed forward. We're continuing active transport in the ascending limb. Okay. So that 400 is still dropping down to 200 as we're moving forward because we are actively transporting solutes, pulling them out of that fluid, pushing them into the medulla. Okay. So this is still keeping that osmolarity of the medulla nice and high. Okay. So this situation continues to exert a pull on the water. Okay. So water is going to continue to then therefore have force pulling it into the medulla. So we're going to see in the descending limb, we keep having water move. Okay. But now, because of that pushing forward of the fluid, different amounts of water essentially are getting pulled. So our previous state, just a reminder before, for descending limb versus medulla, right? We had a bunch of fluid at 300 in the cortex that was pushing down. Okay. And then we had some fluid at 400 here. Okay. And that had been pushed forward a bit. And in our imaginary setup, the whole medulla was at 400. Okay. I'm just going to draw that all out. So what happens here in this situation is that we have a greater difference here up at the top than we do as we're down farther into the loop. Okay. This means that we have more water flowing out at the top than we do down at the bottom. Okay. So we water this water down up at the top a lot. So now we just have a difference between 300 and 350 because of that movement of water. But as we get lower, less water has moved. This has actually gone up because we continue to have solutes pumping on this side, right? And there's not as much of a pull here between the 400 and the 500 as between the 300 and the 350. Cool. So we continue going forward. We're letting this all come to equilibrium in this next image, okay? So now we've started to have a gradient in the medulla, right? So now we start to see that we have lower numbers up at the top of the medulla than down at the bottom of the medulla. Okay. So we have a gradient in the medulla because we had this lower osmolarity fluid at the top with a greater difference from that medulla that we had set up with our active transport. And we continue to run the active transport. Okay, so we have that gradient going down. We're coming to equilibrium in this descending limb with that osmotic gradient in the medulla. And we're having lower values in our ascending limb because it's actively pumping sodium, chloride, potassium out. Okay. So this keeps happening over and over and over again until we get to our steady state in an adult in a normally functioning kidney. Okay. So we can see here that 300 value at equilibrium in the cortex, okay, between our proximal tubule and the osmolarity of the cortex. So we might say they're isoosmotic. Okay, that fluid pushes forward. There's a big pull on it. There was a big pull on it at the beginning, so we had a lot of water flow out. So we have a lower value for our osmolarity near the cortex. But as we dive deeper and deeper, we're coming to equilibrium with a higher and higher number as we dive down into the medulla towards the renal pelvis, which is like our central point, that point where we're going to have the ureters coming out. We have blood flow in. Okay? And at the deepest parts of the medulla, we have values of about 1400 milliosmoles. But we're not done because as we go back up, right, we're pumping solutes out. So we came to equilibrium with this 1400 milliosmoles. But as our fluid starts to push back up, we're actively pumping those solutes out. So our numbers start to go down again. And they actually go down until they are even lower than their original values. So we have fluid in this distal tubule that's at 100 milliosmoles. So this is the key thing to understand and remember, right? So this was our setup. So you do want to know the properties of the descending limb, chiefly that it's permeable to water, and it has water pulling on it. Osmolarity getting greater as we go down into the kidney. And then as we go back up in the ascending limb, we're able to actively transport these solutes, meaning that we're dropping down that osmolarity of the tubular fluid again, at the same time as we are making the medulla highly osmolar, creating this strong pull on the water from the descending. The questions about the medulla and its properties and these properties of the ascending and descending limbs. So we've set up a situation where we exert a pull on water just based on these solute concentrations, okay? Now, there are a couple other factors going on here. This is just the whole picture at once, but we just step through each of those pictures, okay? There are a couple other things that happen here. These are going to be slightly lower priority for you, but I want you to at least hear them. So if they come up in the future, you know some of these words, okay? So a chemical called urea also does play a bit of a role in developing this medullary osmotic gradient. So urea, like urine, right? We're making urine, has urea in it, okay? Urea. This is made by the liver, and its purpose is to eliminate nitrogen in your body. So you are filtering urea out of your blood, came from the liver. Eventually the blood from the liver got to your kidney, and as it passes by, we're taking out that urea, got filtered into the glomerulus, out of the glomerulus into the tubular fluid, just like everything else that's not proteins, okay? It's very water soluble, so it's nice and dissolved here in all your fluids, both in the tubular fluid, also dissolves well in interstitial fluid. However, for it to travel across cell membranes, it needs transporters, okay? So there are three kinds, UTA, UTB, and UTC. I don't really need you to memorize those for the purposes of this course, but you should know that there are these transporters there are different ones in different locations. So here we see UTC transporters in the proximal tubule. We see UTA transporters in the collecting duct. And here we see UTB transporters in our capillaries. Cool. So these are the places that urea is able to move across membranes. Okay. So particularly important bit, I would say, if you were remembering a fact here, is just noticing that two of these urea transporters are down here in the medulla, right? And that's part of why urea is able to add into this medullary osmotic gradient, right? So essentially it's kind of adding to these solutes that are collecting here in the medulla. So adding in to that pull on water. We also want to know that the vasa recta are here. Okay, so great time to retrace our blood flow to the nephron. Okay. So we have our efferent arterial coming in. Sorry, our afferent arterial coming in. I'm getting ahead of myself. Afferent, 2. Coming to our glomerulus. That's our swirling capillary bed. This was where we had our filtration into Bowman space and into the nephron. Then we have our efferent arterial leaving. Okay. So this is a case where we have something leaving, but it's not a vein, it's still artery. Efferent arterial. Okay. We then had peritubular capillaries surrounding our proximal tubule. And these are important because when we were talking about reabsorption, we were talking about things like glucose moving from our filtrate, so from this tubular fluid and going somewhere. This is where they were going, right? So this is how we were reabsorbing. We were moving back into the blood, into these peritubular capillaries. Peritubular capillaries. Okay. The vasa recta here are referring to these straight shot sort of capillary beds. So we have our straight shot down, our straight shot up, and this capillary network in between. Okay. So these are vasa recta. You've seen that word elsewhere, probably as you were talking about abdominal stuff. If you haven't gotten to the abdomen in lab, you will see this term again. Vasa just means vessel, like blood vessel. Recta means straight. So that's why sometimes we reuse this term, because we're just referring to the fact that we have these straight blood vessels. And they tend to travel sometimes related to arcs. Okay. So the vasa recta are just important because they're helping us, they're helping prevent us essentially from dissipating this gradient that we're setting up with our solute movement as we're moving through the loop of Henle and our water movement. And we tend in nature to want to come to equilibrium. So even though we set this up, if you let it just sit still for a long time, it would start to sort of just like wash out and dissipate. But because we have this blood flow continuing to move, water basically moves into the interstitial fluid, but it doesn't stay there. It does go into these vasa recta. Okay. So if you look at the descending limb of the vasa recta, it's at 300 milliosmoles. As it's descending, water does leave capillaries by osmosis and start to enter that medulla. Okay. But in the ascending limb of the vasa recta, we're at a slightly higher osmolarity, but the water in the interstitial fluid can move back into the ascending limb of the vasa recta. So these are different than what our ascending and descending limb of the nephron we're doing. So water can move across both of these. Water leaves and water moves into the plasma. So both of these are permeable to water. Okay. Now it's not super important to me that you know the details of this process, but you do want to know that they're here. You do want to know this blood flow is surrounding the nephron, which is why we can have reabsorption happening basically throughout. Okay. If we were to look at it, something like this is actually tracing through the vasa recta. It looks like the nephron, loop, but it's not. This is actually our blood flow, right? That has that same arrangement because it's just tracing around the edge of our nephron loop or our loop of Henle. So we can see it also has an osmotic gradient, okay? But unlike the ascending and descending limb here, we have two-way movement of both our water and solutes, okay? Because of the movement of the fluid, it's just kind of helping us maintain everything. That's about the level to which we want to understand that. Just kind of know it's there, know it's helping us maintain these gradients. Stuff can move across both limbs. Okay. So now we want to think about our next two pieces of the nephron. So we've just been kind of going through the nephron in order. We had our glomerular capillary, which was part of our renal corpuscle. The other part was our Bowman's capsule, right? Containing Bowman's space, leading us into our proximal tubule. Okay. Now we've looked at our nephron loop. Okay. So we dove into the medulla. Now we're back out in the cortex. We're going to have our distal tubule, and then we're going to move down into our collecting ducts, which actually collect fluid from multiple different nephrons. Okay. So now we're going to have water reabsorption that we actually do regulate and we actually do control. So we had unregulated reabsorption happening here in our proximal tubule, lots of it. That's where we were doing most of our water reabsorption. We saw that that was following the movement of sodium. So we had our active transport of sodium. So we had our sodium potassium ATPase, our sodium potassium pump, moving sodium into this interstitial fluid here, water following along with it, following that sodium. Okay. Now we're thinking about the fact that we can also reabsorb water here and here as we get later on. Okay. But we're going to regulate this. Okay. So now we have to think about the permeability of these epithelia to water. Okay. So in our proximal tubule, our epithelium, our lining of the tubule, was permeable to water. Water could move across that cell layer. Okay. That is not always true in the distal tubule and the collecting duct. We're basically going to be able to turn on and off the permeability of the cell layer to water. Okay. So we're going to do this with our water channels. Okay. So until we think about regulation, we don't really think about aquaporins. Okay. So usually we just think about cell membranes as being permeable to water. But that's actually because usually, unless we tell you otherwise, we have proteins that allow water to go through. Okay. Those are aquaporins. So pores for aqua, water. Aquaporin allow water to move across. Okay. So usually we're just assuming they're there, and that's why water can travel, because they're pretty much everywhere. However, in the distal tubule and collecting duct, they're kind of variable. So they're always in one place. We always have aquaporins in our basolateral membrane, okay? But we don't always have aquaporins in our apical membrane. So now we want to think about a cell with shape. So I'll draw us a new one. Okay. So when we're thinking about a cell here, we're going to think about our basolateral membrane facing blood, apical membrane. We'll draw it like we did before, the one facing our fluid. Okay. And I'll draw our bloodstream here. Okay. So what we're saying here is that we always have aquaporins, and specifically aquaporin 3. And you do want to remember aquaporin 3 versus aquaporin 2. So aquaporin 3, okay, always present down here. Okay. So we're always able to move water from the inside of the cell to the blood if we have the appropriate gradients. Okay. But we're not always able to move water into the cell from the tubular fluid because aquaporin 2 is only present under certain conditions. So aquaporin 2 gets like question marks around it, right? So we have to know whether we have this aquaporin present or not to know whether water can flow from our tubular fluid into this cell. Right. So we saw the difference a little bit between the cells with the distal tubule and collecting duct compared to our cell layer in our proximal tubule before. So differences mainly were that we had these tight junctions. So we have a tight seal here, which means that in order for water to go across and get into the blood, it has to travel across our apical membrane, and then it has to travel across our basolateral membrane. If it can't do both of these steps, then it can't flow into the blood, right? You can't get into this cell, you're not going to set up a gradient where you can get out of this cell either. Okay. So this is why we're going to be able to regulate transport in the distal tubule and the collecting duct, because we're going to control when we are able to do this step. We're going to control the presence of aquaporin 2 to control whether water can come into the cell or not. And overall, that's going to control whether we can move water from the tubular fluid and into the plasma. So whether we are reabsorbing more water or not. Okay. Bless you. All right. So we're going to start out thinking about what happens if we don't have aquaporin 2. Okay. So if the membrane of the late distal tubule and the collecting duct is impermeable to water, okay, and the reason it would be impermeable to water would mean no aquaporin 2. Okay. We're going to have a situation where water can't leave the tubules anymore. So we have our tubular fluid at 100 milliosmoles. So that's pretty low. It's got lots of water in it because we pumped out all those solutes to keep our medulla nice and salty, keeping up that medullary osmotic gradient. If we can't move any more water, we can't reabsorb any more water, it's just going to stay at 100 milliosmoles, right? Nothing can happen to it. The water can't move. Just keeps going out your body, out the ureters to the bladder, you pee it out, okay? Your urine would then have low osmolarity, right? Because it's just going to keep that 100 milliosmoles essentially. And you'd have a lot of it. So the reason for that is your urine, mainly made up of water, right? That's what the fluid is, okay? So if you are not removing fluid, because water can't travel across those membranes, the fluid stays in the urine. So you have a lot more urine, right? You don't reabsorb the water, so it just all stays there. There's lots of it. High volume of urine, low osmolarity. Okay. So water can't leave the tubules because there are no aquaporin 2s. This means we have no water reabsorption. When you then excrete the water as urine, there's lots of water with low osmolarity of that urine. And we have a different situation if we can transport water. The reason we would be able to transport water, do more reabsorption, is if we add in those water channels, if we add in aquaporin 2 on that apical membrane. If we add in aquaporin 2, right, then we have tubular fluid on its way to become urine that's traveling through a very salty medulla, right? We have this strong medullary osmotic gradient that we just set up with our loop of Henle and our countercurrent multiplier, right? So if water can travel, this is a strong pull, right? Water is going to want to travel into the medulla here, okay? So the way that we can have aquaporin 2 is if this hormone, ADH, or antidiuretic hormone, gives your cells the signal to make aquaporin 2 transporters and stick them in the membrane. Okay? So in the presence of ADH, when you have this hormone signal, you insert aquaporin 2 into the apical membrane of the cell. Then water is able to enter the cell. It's always able to exit the cell. Okay? There's a strong pull from the medulla. So we're going to reabsorb a lot of water. Okay, so we're going to end up with more highly concentrated urine. So we're going to see that same sort of situation that we saw in the loop of Henle, right? We're going to start to try to come to equilibrium with this extreme osmolarity of the medulla. So the maximum urine concentration, the most osmolar urine you can have, is the same as our maximum values here at the deepest parts of the medulla. Okay. So the maximum amount of water that gets reabsorbed is going to depend on the length of the loop of Henle, because that's how deep we're going in the medulla, and that loop of Henle is setting up this gradient. So here we can see a comparison of those two situations. In that previous slide, I was just showing you this first one, okay, where we did not have aquaporin 2. So we just keep this low osmolarity in our urine, end up with low osmolarity and high volume because we got lots of water left. Okay. But here we see in the presence of ADH, our hormone, we insert aquaporin 2 into our membranes, which means water is able to flow out. And it does because of this pull from that medullary osmotic gradient. So we have lots of water move. We end up with urine, has all the water removed, so it has low volume, right? Because most of the volume of our urine is water, okay? And we have high osmolarity because we took all the water out. So we did mention before that you do have some obligatory water loss per day, right? In order to excrete your waste products, you do have a minimum water loss of 440 milliliters, okay? Maximum osmolarity of 1400 milliosmoles because of that depth of the medulla. Okay? That's all we're saying right here. So I do want us to take a look at the antidiuretic hormone. We're running up on time. Okay. So we'll pick up here next time. The big takeaway is just going to be when this hormone is present, puts aquaporin 2 in that membrane, and you can move water.